Hi, I am Asisha, and today we are going to talk about. It is the actually continuation of the previous video, where we were talking about DNA sequencing. So in this video, we will move further and we will talk about the second method, which was discovered by Sanger, the Sanger method for DNA sequencing. So what happens in the Sanger method that we add ddNTPs. So we will talk further. So let's move. Dideoxynucleotide chain termination method. So first, anneal the primer to DNA template, usual single. So let me explain it. Then we will go for the reading because there is a lot of reading. So ddNTPs. If you can see the difference, the normal DNTPs have OH at 3 prime end, where another nucleotide will bind with the help of oxygen, and it will make a nucleotide sequence. But if we synthesize in lab such DNTPs which does not contain OH, just contain H. So whenever the nucleotide which is radioactively also labeled add to the reaction, so the reaction will terminate. So that's why it is called chain termination method. So where the reaction will stop, there we will get uh, if it is radioactively labeled, phosphate is radioactively labeled or dye labeled, we will get signal that the reaction is stopped. So we will assume that thiamine is added. So if thiamine is added, we can say that there is in template strand there is a adenine. So first you will understand, first sequence of DNA or nucleotide you will get is that there is a adenine present. So in this way it happens. So let's move for elaboration. First, anneal the primer to DNA template, usually single stranded 5 prime, like GAAT, GT, CC, TTTT, CT, CTAAG. So uh, we are going to make this anneal with the template strand. Then split the sample into 4 aliquots in tubes labeled G, A, T, and C. So these are the 4 reactions. In first reaction or tube, aliquot tube, only G's which contain ddGTP. Okay, and in second one A, only A nucleotide with ddATPs, and so on. So reaction will be like this, G tube wall. So you can see this. When a polymerase clino fragment is added to the tube, synthetic reaction proceed until by chance a dideoxynucleotide is incorporated instead of a deoxynucleotide. This is a chain termination event because there is a lack of OH, but only H is present. Here is a 3 prime H. So since the synthesis for the synthesis of DNA can since the synthesized DNA contains some radio labeled or chemically labeled substrates, the product can be detected and distinguished from the template. You can see the difference between DATP and ddATP. DNA trace synthesis and termination. So this will occur like this. You can see that if OH is present, so O will form bond with the uh, six with the carbon number 6 phosphate of the other nucleotide. So it will make bonding. But if there isn't any oxygen OH group, so it will not make any uh, bond with the another nucleotide's phosphate group. So the termination will occur. And one thing I must say that the ddATP or ddGTP which will be incorporated will also be radioactive labeled or with some substrate. So we will get a indication or some kind of signal that dd NTPs are added. So this is a reaction. You can see that whenever G is added in the G reaction to alicot, G is added, there we will get a signal and the reaction will terminate. So we will get that G is present. The sequencing gel. These products are denatured into single-stranded DNA molecules and run on a polyacrylamide urea gel. The gel is dried onto chromatography paper to reduce its thickness and keep it from cracking, and exposed to X-ray film. Since the template strand is not radioactively labeled, it does not generate a band on the X-ray film. Only the labeled top strands generate bands which would look like this. The sequencing gel, as you can see from this one reaction, the G reaction, the chain termination events produce individual bands on a gel. The chain termination closest to the primer generates the smallest DNA molecule which migrate further down the gel, and the chain termination further from the primer generate larger DNA molecules which are slower on the gel and therefore remain near to the top. So it in the gel, if when we run it, so the smaller will run far from the starting point. So we can say that the smallest will be the smallest molecule which if it is that is ended on G, so we will say that G is the first nucleotide in the sequence. When similar chain termination reactions are run for each nucleotide, the 4 reactions can be run next to each other and the sequence of the DNA can be read off the ladder of bands 5 to 3 prime sequencing being read from bottom to top. So remember that from bottom to top is 5 prime to 3 prime. Manual sequencing, so you will just see that if the smallest is on the bottom is a T, so we will say that first is T, then is other is C, and then the upper one is also C, so we will say that TCC. In that way we will make the sequence of DNA. So it is enough for today's video. We will talk more about the DNA sequencing in the next video, and I will appreciate if anyone suggests some topic related to DNA sequencing or ambiguities. I will try my best to solve it, and yeah, please subscribe to my channel and also like, share and comment. Thank you.